Hello, friends. Welcome to the last topic of hypersensitive reactions, that is type 4 hypersensitivity. We have already, in our previous lectures, we have already discussed mm, uh, the other three types of hypersensitive reactions, that is type 1, type 2, and type 3 kind of hypersensitive reactions. And today we will discuss about type 4. So we have already know that all these type 1, type 2 and type 3 type of hypersensitive reactions, basically there is involvement of antibodies. So all these first three types of reactions had antibodies involvement. Now, but in case of type 4 kind of hypersensitive reactions, uh, there is no involvement of antibodies. Antibodies are not involved in it, and in addition to this there is involvement of T cells. So you can say that it is a T cell mediated immune response. It's a T cell mediated immune response. Now, how do you understand the pathology of this type 4 hypersensitive reaction? Is this type 4 hypersensitive reaction is basically caused by chemical substances which somehow find their way inside our body system. So these chemical substances, it could be like poison ivy, it's a plant, or it could be from poison oak, or it can be any kind of latex, resin, metals, anything can be these any of these molecule can act as your antigens inside the body. So say for example, we will take the example of poison ivy. This poison ivy, basically what it does, it secretes a chemical substance named as urushiol. It produces a chemical substance called as urushiol. एक chemical substance produce बच्चा देमिंग पुलाता urushiol zit. So diri plant pu ngati jaga no present honchin odola hatar da chot jerchin. Lakpe ka ngati tash shana ka tuji shet ngati lakpa merba longet और ngati lakpe ka chishet blisters shet मतलब वो छोटे छोटे हमारे यहाँ पे बॉडी के ऊपर क्या बन जाते हैं ngati दाग चुक्षचा. So say for example, this urushiol, it comes in contact with our skin. This is your epithelial lining of the skin. This urushiol, I'm trying to draw it in red. It comes and it enters into the epithelial cells. It goes inside the cells into the epithelial cells. There it binds with small protein molecules of these cells. After binding with small molecules, now I would try to draw here. These are the proteins and these are the chemicals. Now these chemicals or the urushiol, it binds with the proteins. So because this chemical, it cannot be act as an antigen. Why? Because of its smaller molecular size. We have already discussed in characteristics of antigen that there should be a proper molecular size so that it can act as a pathogen. Now this antigen, this chemical substance and the protein, now in complete, it they can incomplete act as an antigen. Individually, they can't act as an antigen. Basically, small molecules which are not immunogen but are antigen are called as haptens. Now, this whole complex molecule is an antigen and it can now cause immunological responses. So, what happens that it is being now taken by our cells that is called as our antigen presenting cells. So say for example this is an antigen presenting cell. Example we can take as of macrophages. So this macrophage, they are antigen presenting cells. They will present these molecules along over their cell surface along with MHC2 molecules, because it is an outside pathogen. It's not a viral particle, it's an outside pathogen. That is why it is being represented over the MHC molecules along with the small proteins of the cell with the help of MHC2 molecules. Fine. So now this is our antigen presenting cell. I will try to draw it again here. This antigen presenting cell with their MHC molecules, and it has the antigen over its surface. Now it comes in the circulation and it finds a way. It finds a way and finally it counters T cells. It counters T cells. These T cells interact with the antigen presenting cells with the help of their T cell receptors, which we called as TCR. They come in contact with these TCR molecules, and because of this connection and even co-stimulation, a connection and co-stimulation, now this releases certain interleukin molecules, proteinaceous soluble molecules like IL-12. Because of these IL-12 molecules, this naive T cell, which has not been differentiated into subtype of T cells, this naive T cell means it has not been differentiated, मतलब इसका अभी कोई identity नहीं है इस T cell का. So this naive T cell, it will now differentiate because of the activity of IL-12 into another subtype of T cells, which is called as T helper cells. So this is your Th1 type of cells. So this Th1 type of cells, now it again recognizes the antigen presenting cells and it will start producing interferon gamma. What does this interferon gamma do? It will again activate further macrophages. It will further activate the macrophages, macrophage activation. It will further activate these macrophage molecules. So these macrophage then activates it, can act as antigen presenting cells in order to clear those pathogen or the uracil molecules entered through the skin surface, or they releases reactive oxygen species like nitric oxide and oxygen free radicals, and they also releases lysosomal enzymes. So in addition to these, macrophages can also secrete tumor necrosis factor, that is TNF alpha or IL-1. Now what does these two chemical substances will do? We have the endothelial lining. This is endothelial lining of the circulation. So we have endothelial lining of the circulation. As this TNF alpha and IL-1, it interacts with this endothelial lining, they produces receptors. They produce receptors over these endothelial lining. Say for example, some kind of receptors are being produced over here. Now what these receptors will do, they will recruit more kind of leukocytes. Circulation mein when there are leukocytes, they are being attached and they are being taken to the interstitial spaces where there is infection, that is chemical substance have entered. So more leukocytes are recruited to this area. So more inflammatory responses obviously will occur. So this is one way of how macrophage activation with the help of T cells occurs. Now another kind of, say for example, this naive T cell, it can differentiate into another kind of T helper cell, that is by the activity of IL-6. Say for example, it releases interleukin 6. If macrophage, when it comes in contact with naive T cells, it releases interleukin 6. This interleukin 6 now differentiates this naive T cell. Instead of Th1 cells, it will now differentiate into Th17 cells. So this is your Th17, and this Th17, what it does, it releases IL-17. It releases IL-17. When IL-17 is released, what it does, it activates neutrophils, another kind of WBCs. Now these neutrophils, they can release certain kind of enzymes, lysosomal enzymes they will release, or they can also release reactive oxygen species or the lysosomal enzymes. Similarly, what happened with that of macrophages, so lysosomal enzymes are also being released by these th 17 type of cells. So the whole picture is something like this, that first the uracil enters into the system, and it is then clubbed with the kind of cells uh, which are antigen presenting cells, and whole of the cycle goes on like this. Now even these macrophages can also come in contact uh, with this naive T cell, can also be then differentiated into Th2 kind of cells. T helper type of cells, they are also being differentiated. This T helper cells, they will now release IL-4 and IL-5 subtypes of cytokines. Now what they will do, they will recruit eosinophils over that area. They will recruit eosinophils, and these eosinophils now releases proteases, various kinds of digestive proteins, and finally leading to tissue damage. So the final pathophysiology is that there is occurrence of tissue damage in case of type 4 hypersensitive reactions. So this is how it goes. And now this type 4 kind of hypersensitive reaction is also called as delayed type of hypersensitive reaction. This is also called as delayed type of hypersensitive reaction or hypersensitivity. Why it is called as delayed type of hypersensitivity is, we'll try to explain here. Basically there is activation of various kinds of immune cells, and after that there is recruitment, more kind of immune cells are being brought over the area, and then finally there is a response generation in the form of inflammation or redness or rashes over the skin. So certain examples, very important examples in this type of hypersensitivity is, we have vesicular dermatitis. So it is a disease of our skin, जो ngati skinny disease shikin, जहाँ पे ngati skinny layer पे खतुई का जब कोई केमिकल चिन्हबो ngатang, अगर उधर ने हमको ngатang कांटेक्ट आयो ना का, तो उसके ऊपर छोटे छोटे ब्रिजल्स बनते हैं, ठीक है. विच इंग्लिश में आता है दो जर्सिन ब्लिस्टर्स, द ब्लिस्टर तंचिन दो लाते जर्सिन वेजिकुलर डर्मेटाइटिस. अब दो चेल दानिन जरने का तो चाजमना उदानु ngata धीरी स्नल लाते सिल्खनी इम्यूनोलॉजिकल रिस्पॉन्सन गामा उदानु ऑकर छाना, ठीक है जी. So this vesicular dermatitis, it again takes some kind of time, say for example it occurs in a time duration of 24 to 72 hours, or you can say 1 to 2 days, एक से दो दिन के बीच में तेरी एलर्जी रिस्पॉन्स आता है उनसे. So that is why we call it as delayed type of hypersensitive reaction. So another example in this type of hypersensitivity is TB skin test. So this TB skin test, what they do basically, they inject tuberculosis, Mycobacterium tuberculosis proteins beneath the skin. Now, the moment it is being injected, if there is a response after 1 to 2 days, that means the test is positive, and you can say that the person is either being given TB vaccine, or it has been already infected, already infected by tuberculosis at certain time in the past. Another example is contact dermatitis. So this contact dermatitis basically it happens because of, say for example, metal allergy. Classic metals are like nickel. कुछ लोगों को होती हैं, इवन गोल्ड ज्वेलरी अगर खर्ची तक नगद हो, उनको कुछ दिन के बाद लगता है कि मतलब ब्रप्सियाँ हो रहा है, इचिंग हो रहा है, रैशेस फॉर्मेशन हो रहा है. दैट इज़ बिकॉज ऑफ type 4 kind of hypersensitive reactions. Basically that person or that guy is allergic to certain kind of metals. Now in addition to T helper cells, there is involvement of cytotoxic T cells. Also, in case of cytotoxic T cells are also involved in type 4 kind of hypersensitive reactions or type 4 hypersensitivity. So, say for example, we will take type 1 diabetes. So in case of type 1 diabetes, what happens that these T cytotoxic cells, they will start attacking, they will attack beta cells of pancreas, and because of this the metabolism of sugar is being hampered. That is why the person is called as type 1 diabetes. Another example where T cytotoxic cells are involved in hypersensitive reactions, these are also basically autoimmune responses. Hashimoto's disease. So what happens in Hashimoto's disease is basically, T cytotoxic cells. These T cytotoxic cells, they attack thyroid follicular cells. They attack thyroid follicular cells, leading to hampering of the thyroid glands. Now, in addition to T cytotoxic cells, T helper cells also are responsible for Hashimoto's disease. T helper cells, when they activate macrophages, these macrophages then starts killing the thyroid cells. How they start killing the thyroid cells is basically they will release certain kind of granzymes. These granzymes, they will finally lead to apoptosis in these thyroid cells. So another example could be graft rejection. The graft rejection, involvement of graft rejection is basically by the involvement of T cytotoxic cells. the graft which is placed are being rejected. सो द ग्राफ रिजेक्शन पता ना मुझे ना से फॉर एग्जांपल एक बंदे को एक्सीडेंट हो गया दे मिला एक्सीडेंट छना का तो कोई पार्ट की जगह चिकन है शेक चाहते यहाँ जगह चिक स्पेयर या किसी दूसरे बंदे से लाता है जबकि ए बंदे का नखो मना चीज़ चिंजन का तो उसका जो बॉडी के सेल्स हैं वो ए मी फिलाची सचिन वो क्या हो जाता है इट एक्ट्स एज अ फॉरेन तो उसके अगेंस्ट में हमारे पास इम्यून जनरेशन होता है immune generation चनाता है ग्राफ रिजेक्शन समटाइम्स ngati बॉडी के पार्ट खेर से अगर उदू एक्सेप्ट मबन अगर तो दैट इज ऑल्सो कॉल्ड एज ग्राफ रिजेक्शन इट इज ओनली बिकॉज ऑफ hypersensitivity type 4 kind of reaction and because of these TC cytotoxic cells the graft is unable to be accepted by the body. So hopefully we have completed all the topics in this chapter. Till the time we will come with another chapter with new topics. I would like to say goodbye to all of you. Be safe and please study well. Thank you very much.